Now we're going to open it up to questions. If you have a question, you can raise your hand using the WebEx controls, and we will unmute your line, and you can ask it over the phone, or you can send a chat to myself or through the Q&A box as well. So, Janet, we already have a question come through, and it's regarding, this is from Sangeeta Kaur, and she was wondering how biomarker identification is affected by the use of drugs in the patient samples that they've been prescribed, and if it's always essential to include untreated patients.
Sangeeta? Yes, Sangeeta. Thank you for the question. That's an excellent question. And in fact, when you're, as you're advancing your biomarker panel down the path, there's actually in the establishing analytical performance phase of your study, there are protocols for understanding, they're called interference. So, for instance, if you're collecting a nasal sample, you really need to be able to annotate if the patient, for instance, is using Afrin in the morning. And one of the things that you can do, and I'd love to talk to you about the details of your study, one of the things that you can do, and it's not always possible, is but you can make it one of the requirements of the cohort selection that people haven't used Afrin or, like a nasal spray for 3 days prior to collecting the sample. Now, if that's not possible for the type of study that you're doing, I mean, you've got patients who are being treated. Obviously, there would be some ethical considerations with respect to requesting that people stop treatment. If you can do it, it is incredibly valuable to collect samples from people who are treatment naive, that is, people who have not been treated, so that you have a good understanding of the impact of that treatment on the samples that you're collecting. There's a second part of this, and that is even with the patients or the volunteers who would come in to donate a sample who aren't on the treatment in question, you really want to talk to them about other treatments that they may be taking, medications or special diets that they're on. And again, that gets to the lab metadata, because if you see some subclustering within a subclass that you're really struggling to understand, it's really helpful to be able to go back to your lab metadata and say, oh, wow, these are the people who are smokers. And, their lung microbiome is different, and it's not due to, the treatment per se, but it's due to something else that's being introduced into the lung. So it's a great question, and it's great that you're thinking about the controls in this way, and to establish a robust biomarker panel early in the process. These are the types of things that you should be thinking about. So, love the question. Thanks.
Okay, great. Thanks, Janet. We have another question here about the structure of the 16S gene and which part to analyze in order to effectively distinguish the phylogenetic diversity.
Okay. Oh, that is a great question. And in fact, that's a question that I anticipated might come up. And I have a slide. Can I show the slide? Do I have this? Okay. I have a slide in here. You're right. Microbial studies often use the small subunit 16S ribosomal RNA gene for phylogenetic assignment. And in most experimental designs, either the entire 16S gene or portions are analyzed by sequencing or hybridization. So the trick here is selecting enough of the informative region of the 16S gene for accurate taxonomic assignment. It's critical. One way to think about this is all of these bacteria have evolved from each other. So they're all very related. And there are regions of the 16S gene which are unique and informative, and then there are regions that have that are shared. So this figure illustrates the reliability of taxa assignment across multiple short stretches of 16S sequence. Correct assignment is dependent on the location and length of the region, as well as bioinformatic methods employed. Sequencing errors can further confound this analysis. In our method, we address this using an approach that interrogates the full length of the gene for maximal discriminatory power, and we also build in internal controls that help us when we're doing the bioinformatics and taxa assignment. So great question. Thank you.
Okay, great. Another question here regarding the number of samples for studies with mouse strains. Could they be pooled?
You know, that is a question that I get often. And I understand the desire to do pooling, but there are also individual differences between even litter mates. And some really nice work was done years ago by Bruce Conklin and other people looking at litter mates. And although they're genetically similar, what we now know due to all of these robust whole genome technologies is that even identical twins aren't identical. So my response is it depends on the question that you're asking. And I'd love to talk to you in some detail about your proposed study and the question that you're asking, because there are hormonal influences in utero in litter mates that affect the microbiomes and even development of the individual mice in a litter. And so, for instance, if you have a male mouse that's positioned between two female mice in utero, that male mouse is different than a male mouse that's positioned between two male mice. Now, that may not matter for your study, but it might. So, it just depends on the question that you're trying to address, and if the differences within the litter mates, within your groups, could confound your interpretation of the data later, and that you'd be losing information by pooling. So call me and let's talk.
Okay, great. Thanks, Janet. I think that's probably kind of nearing the end of our time today. If you have any other questions for either Janet or for us at PhyloTech, you can contact us. The email address is listed on the slide here, info@phylotech.com, and we'd be happy to answer any follow-up questions that you may think of later today. I want to thank you, Janet, for a great talk. It was really, really interesting. And thanks to everybody on the call and who joined today. We appreciate your time. And thanks, and have a great day.